Okay, this is part two of the project preparation material. I'd finished by talking about loss and gain of function, and I also want to introduce a couple of other terms as well. Um, and so there are two different ways to introduce mutations to genes. Um, and reverse genetic approaches, which are underlined here, are when you have a particular gene in mind, you want to knock it out. You want to do something like the RNAi method, which I'll be talking about a little bit later in the semester. And so you target the gene and you basically reduce its expression. And there are several methods to do this. You may know some methods from 357 that we probably won't cover here. I don't want to have too much. A little overlap between courses is good because it shows you how they're connected, but too much it will end up being a little bit boring for you. So a forward genetics approach is when you're studying a process rather than a particular individual gene. And so, and this is how before we had a sequenced genome and so on, we did this way all the time. And it's basically you make lots and lots of mutations to do your screen, like the genetic screens I've been going over in the previous lectures. And then eventually you isolate strains that have mutations of interest, and then you look at the phenotypes and whereabouts they are in the chromosome, and eventually you figure out which gene is altered. It's a lot of work compared to reverse genetic approaches, but the advantage of it is sometimes you are interested in how eyes develop, and the genes that you think are the important ones turn out not to be, or at least they're not all the genes. There may be additional ones that are contributing, and you don't know based on what you know about sort of particular genes sort of sequence and the protein you think it makes and so on. So you can get more information with a forward genetics approach over a whole process. If you are wanting to do something very directed, and you're really only interested in one gene, then using a reverse genetic approach makes more sense. It's more efficient. You can get right at that particular gene. Now, our screen is a forward genetic approach. We are going to make mutations by mobilizing a P element, but I've introduced a shortcut that makes the work much less. When I designed this experiment, I thought we might have to look at thousands of flies before we got mutations. And I think the students, because we had a catastrophic failure in the incubator, the students were only able to do something like 100, 150 or something crosses, and we got a mutation within those 150. So it was a very good shortcut. Okay, and I've put this in your notes so that you can just write down a few thoughts about the two different approaches. I would suggest writing in your own words what each is, how they differ, and then what you think the main advantages of each type are. Okay, so we decided, we went on to Flybase. Flybase is a wonderful resource. Every few years they change exactly how you find certain kinds of information, but if you click on DMAP1, this is it here. This is the gene. And this is about 2,000 bases in length. So it's this is a very expanded version. We normally see the maps a little bit smaller. And every single one of these notations down at the bottom here shows you the exact position of particular P elements. And some of them have, I talked about engineered P elements. Some of them have different features on them that allow you to do a variety of different additional things. Find the expression pattern of the gene that your P element is inserted into, and stuff like that. So the one we picked is this one at the bottom here, because you can see these little lines, which I'm pointing to with my finger. Oops. and it's a little bit closer. So it's about 200 nucleotides, so just 200 bases between the P element and the beginning of the DMAP1 gene. There's another gene over here, CG33785. It's known by number. It doesn't have a name because not much is known about it. And these are they appear to be separate genes because the blue is the coding part. That's the amino acid sequence part. And so it's the same transcript, but there are two different proteins you can get from it. And very little is actually known about this gene. And this is transcribed in this direction. DMAP1 is transcribed in the opposite direction, and there's a very tiny little region between them. Here, the big boxes are the amino acid sequence. The little dashes, those are the introns. These are introns. And the purple part at the beginning and end, those are the untranslated parts. So they form part of the transcript, but they are not translated into protein. And so just so that you have a sense of what you're looking at here. We decided to use a stalk that had a P element right beside the gene as close as possible because we wanted to make a deletion originally. And I just started calling this stalk P star because I didn't want to write all this every single time I wrote it out when I was making crosses and so on. It turns out that you can make flies that are homozygous for this P element insertion. And there's no phenotype. They're not sterile. They're definitely not lethal. It's not a lethal combination. And we don't, when we look at the flies, see anything odd that we think is a visible phenotype. And so there's multiple reasons that may be the case. So option one, the P element is sitting there and it's affecting this gene, which is not an essential gene, but somehow it's not affecting this gene, right, which is an essential gene. That's one possibility. It's maybe not affecting either gene, which seems unlikely because you probably can't see it very well, but it's actually right just barely inside those actual genes. Yeah, so you would expect that it's actually having some influence on that gene, but we don't know that for sure. So that could be one option. It's not affecting either of them. It's not making any difference to how they get transcribed, and so therefore they're making their protein, and the flies are perfectly fine. Or it could be affecting DMAP1 and reducing or eliminating its expression, and DMAP1 may not be an essential gene. So there's lots of different interpretations, and so sometimes when you have a P insertion into or affecting an essential gene, you have homozygous lethality. So you would have P star over some balancer chromosome. That is not the case. Okay, so that means we still have a question mark. We don't know. We don't know if either of these are essential genes, and this is the one we want to find out about. Okay, so here's the idea. You've got our P element. This is not to scale because this is about 9,000 bases, and this is about 2,000. And this one is something like 14 or 17,000 or something like that. It's really big. And it's normally found in invertebrates. It's normally found in tendons. It's a tendon specific protein. So this is what we're trying to do. What we want to introduce a wings clipped P element, and we want to see the P element move. And in some of the chromosomes that we test, we're hoping that it's going to insert into the P element. Now, one of the advantages of this is that we have, Sandra's lab has used a primer that is specific to the edge of the P element, and another one that's specific to DMAP1 to do PCR amplification to just confirm, because sometimes the stalk could be mislabeled. And so you think you've got a P element stalk, with the P element right beside DMAP1, but in fact the label is wrong and there's a P element, but it's somewhere else. I have occasionally wasted a few months on a stalk that I received that was not correctly labeled. and it took me time to figure that out. So there's a certain band size. So after PCR, you make many, many copies of just this sequence. And it's about, I'm trying to remember, I think it's 1.9 kb, so around 2,000 bases in length. And so just think about if we manage to get that P-element to insert, what would happen if we did a PCR using those same primers? Okay, that could come into the end of your paper on follow-up. What are we going to do if we find a mutation? What kind of ways can we try to make sure that the P-element is actually inserted in DMAP1 before we spend a lot of time studying that mutation? Okay, so there's no way for us to make the P-element go into this region, but there is a way that we can select for a DMAP1 insertion. Or select for chromosomes that have insertions with a greater chance of finding DMAP1 inserts. So think about, if you think about chromosome 2 that's got around 8,000 genes on it, we don't want the P-element to insert all over and then to find hundreds and hundreds of chromosomes that are homozygous lethal and then have to test each one to find out if it's got an insert in DMAP1 or not. We can use the deletions we made in a previous class, which I will explain about in a minute, to select for the ones we want. Okay, so here's what we're going to do. The first step is to mobilize the P-element. That means we cross P star and we combine it with a P wings clipped, because this is a non-autonomous element. It can't move by itself, and this one can't move, but it can make transposes. Remember, we combine those two types of mutations so that we can control just a little bit of movement and then collect the chromosomes. Okay, so we'll generate a number of potentially mutant chromosomes. It turns out we didn't need nearly as many as I thought we did to find a mutation. And then we cross individuals that have maybe that insert over a balancer to one of the deletions we made in the class. And the deletion is missing about 10 chromosomes. I'll show you about that in a minute. So we've got the P insert over a balancer, and we're crossing it to flies that have the deletion over the balancer. And then we're going to ask, can we get flies that have this P insert in combination with the deletion? Okay. If there's no DNA in the DMAP1 region here, and this has a P-element insert, we're hoping that we're going to see lethality. We're not going to see those flies that don't have the balancer. Or if we see them, they're sterile, because we have some evidence that making mutation in DMAP1 should lead to sterility. Okay. This is referred to as failure. If this is lethal or sterile, that is called failure to complement. And I have, some of you will know what this means and some of you won't. So I put a optional supplementary video that explains what complementation is and how it works. We call this selecting for lethality over the deletion, meaning when over basically is the way we draw it with this on top and this below. So the P-element chromosome is over the deletion, but we really mean in combination with. That's a shortcut. And what that means is it's going to save us a lot of time, because we're only going to identify mutations potentially in maybe 8 to 10 genes instead of the 8,000 that are found on the whole chromosome. So we do much less work. The deletions that we made used that P star mutation, and we selected, and you don't need to know the details except that sometimes when a P-element moves, there is a mistake that is made. It doesn't cut out nice and neat, but instead when it cuts out, it makes a little deletion in the region right beside where the P-element was. So if you can do this on a large scale and make lots of different deletions, all of which start at the point where the P-element was, and then remove small sections of DNA to varying lengths. And these lengths can be anywhere from 100 bases, really short little ones, to maybe 1 to 200,000 bases, a really big deletion, removing lots of genes. And so we made two different ones. One is from 2014. This was the 14th fly that was tested by group 10 in the lab, and K815. So I always did a bunch alongside. It increased the amount of chromosomes we could get through, but it also, I was there doing the work along with the class because I'm part of the team, right? So K is me, 8 is the 8th fly that I found, 15 is 2015, and brown is just a mutation that's on the chromosome. It's a recessive mutation, so if you were actually doing the crosses, you would not see brown because it's recessive, but it causes the eyes to be brown. Sandra's students, a couple of her research students, did some PCRs and other things and discovered that around 27,000 bases were removed. So it didn't just, what we really wanted was a little deletion like this that just only removed the DMAP1 gene but had all the rest. But instead, these deletions remove DMAP1, LRT, this gene and this gene, which may or may not be real, but are inside the LRT gene, a couple of tRNA genes, this, and this. So approximately 10 genes. And then there's a big gap. There's a big section of the chromosome here that doesn't have any genes on it. So we're pretty sure that these are the only ones we have to be concerned about. And they're maybe a bit bigger than 27,000, but that's the approximate size of the deletion. So if there's an insert in any of these genes, we might see lethality in combination. But this is still a lot fewer genes than the whole chromosome, right? So it's a real shortcut. And so the idea is if there's a P element that is inactivating DMAP1 on one homolog that, say, came from one parent, and then there was a homolog of chromosome 2 from the other parent that had a deletion, then you're not going to have any protein from here, and you're not going to have any protein from there. And if the genes are essential, then basically you're going to have lethality, right? Or if it's not essential for life, but it's needed for fertility, then you're going to have a bunch of flies, but they're going to be sterile, right? And so I've posted a couple of questions here and there, which if you're following as we go through the explanation, hopefully you'll be able to answer these questions. Okay, so that's the general approach, and then I am going to, for the last recording, just go carefully through the crosses with the genotypes added, and that should help you think through the screen. And then when we go through it in the lecture, I will actually do the same thing, but we'll have places where we stop and I ask you some questions to try to ensure that you're still on board and that you're kind of still following the procedure. So just like any local hop kind of, or any kind of P element screen, you basically mobilize the P element, then you select a large number of chromosomes that you're going to test, and then each of those individuals has to be tested. In our case, though, we don't have to make the chromosome homozygous. That's a lot of extra work. Instead, we're crossing them to the deletion stock. And then if you have any chromosomes where you've got a lethal combination between your P chromosome and your deletion chromosome, then we're going to keep those and make a balanced stock. Okay. And I will also have a worksheet for you to do in the lab in a couple of weeks that takes you through all the genotypes, and you write out all the progeny that you get, which ones we're picking, how can we tell them apart from the other ones, where do we do en masse, where do we do individual, and so on. And by going through it multiple times, it'll start to make more sense. The first time is going to be awful. I understand that right now you might be feeling really confused, but if we just go through it step by step a number of times, it gets easier each time. Okay, and that's enough. I will pick up here next recording.